Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome back with your tutor Dr. Shehla Aman, and today we are going to start with the histology of the male reproductive system. So objective of the today's class are to understand different histological feature of the testes and to understand the different stages of the spermatogenesis. So a little bit about the gross anatomy of the male genital system. Male genital system consists of the pair genital organ, that is testes, and present in the scrotal swelling, and behind the testes epididymis is present, which receives spermatozoa, and then spermatozoa ascends through epididymis into the ductus deferens or vas deferens, and this vas deferens passing through the prostate enter into the penile area, and then it ejaculates its content outside through the urethra. So histologically, the testes are covered by the thick fibrous coat, which is the tunica vaginalis, and this tunica vaginalis is derived from the peritoneum. Inside the tunica vaginalis, there is another fibrous coat, which is tunica albuginea. This is tough, it contains collagen fiber, and inside is the tunica vasculosa, and this vasculosa forms the septas in the substance of the testes, dividing the glandular element into the lobes and the lobules. At the hilum, you can see the testes are present in the mediastinum testes, then the straight tubules following into the efferent ductules, and then to head of epididymis. Body of epididymis, tail of epididymis continues as the vas deferens. So testes are covered first by the tunica vaginalis, and then next to the tunica vaginalis is the tunica albuginea, which is the tough course, and innermost coat is the tunica vasculosa, which is dense connective tissue having collagen fibers. The lobuli testes are the seminiferous tubules which are present, and between the seminiferous tubules there is interstitial connective tissue, and this interstitial connective tissue containing cells called the Leydig cells. From the seminiferous tubules, the straight tubule or tubuli erecti started which transport the spermatozoa into the rete testes, which are present in the mediastinum testes. From the rete testes, the spermatozoa enters into the ductuli efferent or efferent ductules, and then into the head of the epididymis. So what about histology of the testes? The testes are formed by the different sections of the seminiferous tubules. These seminiferous tubules are highly coiled structures, and they are lined by the specialized type of the stratified epithelial cells. Two types of cells are basically present in the testes. These are the spermatogenic cells and the Sertoli cells. Surrounding connective tissue around the seminiferous tubules is called tunica propria. The seminiferous tubules are composed of spermatozoa located at base of the epithelium with the large rounded nuclei, and the Leydig cells are also called the interstitial cells of the Leydig, are present in interstitial spaces, also called the tunica propria. These are large polygonal cells, strongly acidophilic, having characteristics of the steroid secreting cells, which is well developed smooth endoplasmic reticulum as well as the mitochondria. They secrete the testosterone. These cells are Leydig, also having the lipochrome pigment, and the crystals of the Reinke. The functions of the crystals of Reinke not known yet. Also found within the seminiferous tubules are the large Sertoli cells, also called the sustentacular cells, also called the supporting cell. These are large cells, relatively pale staining and irregularly shaped, having prominent nucleolus within the Sertoli cells nuclei. These cells primarily support and nourish the germ cells in the testes with long apical cytoplasmic folds. They contain testosterone and FSH receptors. Also, another very important point is that these Sertoli cells have tight junctional complexes between them, and because of these tight junctional complexes, they divide the cells into two compartments. The one is the basal compartment, which are having having the spermatogonia in different stages, and other is the adluminal compartment with the advanced stages of spermatogenesis. The cells of advanced stages of spermatogenesis are present. The Sertoli cells, also acting as a supportive cells, have recesses, and all these cells during different phases of spermatogenesis are embedded in the recesses of these Sertoli cells. So spermatogenesis consists of the different phases. The first is the spermatogonial phase, second is the spermatocyte phase, and third is the spermatid phase. So in spermatogonial phase, we can identify different types of the cells. These are the type A dark cells, type A pale cells, and type B cells. The type A dark cells divide mitotically, and they maintain the pool of the spermatogonial cells, and these have the finely granular chromatin in their cytoplasm. Some of the type A dark cell then form the type B type A pale cells, and then type A pale cells starts the process of the meiosis and form the type B spermatogonia. These type B cells have dense chromatin inside their cytoplasm. So spermatogenesis is development of the sperm from spermatogonia, multiple divisions, two meiotic divisions, meiosis first and meiosis second, and spermatogenesis is maturation of the spermatid into sperm. So what happened? The spermatogonia type A divide mitotically, and some of them also maintaining the pool of type A spermatogonia, and some of them become the type B spermatogonia. Spermatogonia type B again mitotically divide to form the primary spermatocyte. They migrate to adluminal compartment, and then they start the first meiotic division. And before first meiotic division, they replicate their DNA so that the cells entering into the first meiotic division have 4 and double the amount of the DNA. After the first meiotic division, secondary spermatocytes are formed, and when secondary meiotic division completes, spermatids are formed, and then next phase is started, which leads to maturation of the spermatozoa. So the next phase is the spermatid phase after the spermatogonial phase, and in spermatid phase a rounded shape structure is then transformed into proper shape of the spermatozoon. So in this phase, first is the Golgi phase. In this Golgi phase, proacrosomal vesicles are initially formed, and they form the acrosome. These acrosomal vesicles are formed from the Golgi apparatus. At the same time, the centrioles migrate and come to lie to the posterior pole of the nucleus in juxtanuclear position, and this distal centriole starts to forming the exoneme of the flagellum, that is the doublet of 9 with the central microtubules. At the same time, the second phase start, and this second phase, which is also the cap phase, in which the acrosomal vesicles come to lie on the anterior pole, and they cover the anterior pole of the nucleus. The third phase which occurs, so the third phase occur in which is called the acrosome phase, and acrosome phase, there is the reorientation of the cell in such a way that anterior pole faces the recesses in the Sertoli cells, and the flagellum faces the lumen of the cells. After this phase, the last phase occur, which is maturation phase. In this maturation phase, the extra chromosome is extruded out of the body as the residual chromosome. Centroli completely forming the flagellum and the centriole, which are present now near to the nucleus, forming the connecting piece. Exoneme is formed, and the mitochondria arrange themselves and spirally arrange themselves around the exoneme, forming the middle piece. So in this you can see this is the neck piece or connecting piece, where you can also see the centriole are present. Then is the middle piece in which in the center exoneme is present, and helically wrapped mitochondria are present. Then the last piece, which is principal piece, and then the end piece. So these are the different phases of the spermiogenesis. Next, what is the sperm? The sperm is total maturation time is 64 days from first meiotic division to spermatogonia A. It has the head having acrosomal enzyme, the nucleus, the connecting piece having pair of the centriole, middle piece having mitochondrial sheet and outer dense fibers and exoneme. Also the principal piece which is having ribs of the fibrous sheet, outer dense fibers and exoneme. So this is the histological slide of the testes with H&E staining, and in this slide you can see a section of the seminiferous tubules. This is the epithelium of the seminiferous tubules, and you can best appreciate that this is the specialized type of the stratified epithelia having two different type of cell. One is the Sartoli cell and the others are the spermatogonial cells. So spermatogonium is present in the basal compartment, and after developing into spermatocytes they move to the adluminal compartment and then they form different stages. These are the spermatid which have reoriented themselves in such a way that their anterior pole is embedded in the recesses and posterior pole having the flagellum faces the liminal side. Another H&E stain section, you can see these are the sections of seminiferous tubule, and this is the tunica propria or interstitium having the polygonal Sartoli cells which are present in the interstitium. So testicular interstitium containing blood vessels, lymphatic vessels and Leydig cells. Leydig cells responsible for production of the testosterone and stimulation of the spermatogenesis, maintenance of sex glands, sebaceous glands regulated by the luteinizing hormone of the hypophysis. So this is another diagram for the testes H&E stain section, in which you can appreciate the specialized stratified epithelia. In the center you can see bunch of spermatozoa. This is the tunica propria, also called the interstitial tissue, which is having the Leydig cells as well. Another important thing that some smooth muscle cells are also present in this, which are also called the myoid cells. Higher magnification of the testes and the seminiferous tubules, and in which you can see the tunica propria. Another higher magnification in which you can see different stages of the spermatogenesis. So sperm maturity and transport pathway. The sperm which are formed in the seminiferous tubules, then they travel through the straight tubules into the rete testes, and from the rete testes they enters into the ductuli efferent or efferent ductules, and here through the efferent ductules they enter into the head of the epididymis, and then passing through the head of epididymis, the body of epididymis and the tail of epididymis. Then they enter into the ductus deferens, and then through the ductus deferens into the ejaculatory duct. So tubuli recti at rete testes. These tubuli recti are the having only the cuboidal Sartoli cells in their lining epithelium, and there are no spermatogenic cells. In the tubuli recti are straight tubules. These are surrounded by the lymphatic channel, the blood vessels and the Leydig cells. So here are the references for today's lecture, that is Histology, the text and atlas by Michael H. Dross. And thanks a lot dear students for your attention. Till the next lecture, Allah Hafiz.